Good morning. So, my name is Ashutosh Kumar and I am professor at the Department of Bioscience Bioengineering, IIT Bombay. I am a structural biologist, use NMR as a primary tool to elucidate structure, function, dynamics of a protein. But recently we got interested in biologics and biosimilars. So, today I am going to show you how NMR spectroscopy can be used for in-formulation state higher order structural characterization of biopharmaceuticals. So, let us start. So, as we know, like I will just briefly mention to you what we do in our lab. So, we develop NMR methods in solid as well as in liquid. In solid, we are trying to detect on protons because biopharmaceuticals cannot be isotopically labeled. So, we have developed a method where we can detect biopharmaceutical at natural abundance using proton detection by spinning at moderate speed of about 30-33 kilohertz. This is one of the pulse sequence that I showed you here, and then we use liquid state NMR essentially for some of the like receptor binding. That is an important aspect, how a drug molecule binds to a receptor, what are the epitopes involved in that. So, here we have developed a method which can be used for receptor binding elucidation, and then we also basically use this NMR spectroscopy on molecular complexes, like we are working on protein intrinsic dynamics and recognition, like this is a multi-protein complex ubiquitin E3 ligase where you employ some of those methods. We are interested in histone tail dynamics in nucleosome and also a structure toxicity relationship of amyloids. That is about the research that we do in our lab. But coming back to biologics. So, what are biologics or biological products? So, if you look at a textbook definition of this term biological product, it means anything like a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component derivatives, allergic product, protein or analog. Whatever coming from the living sources that can be broadly classified as a biologics. It can be a small peptide based drug or a big protein based drug or whole virus. Many of these are classified as biologics. Now, a biosimilar is a biological product that is highly similar to already marketed drug, or this is called RLD, reference listed drug. It should not have any clinical meaningful difference and it should match with FDA approved or EMA approved reference drug. Those are called biosimilar. So, as you know, these are derived from the living sources, either cell or tissue or whole organism. So, these are complex molecule. If you compare a small molecule based drug like say which is used for treating say pain and all those with atorvastatin, this is also a small molecule based drug. Both of these are about 500 like 151 kilodalton and this is 558 dalton. But when we move to biological drug, something like filgrastim, it is a like about 19,000 dalton. So, you can see how many folds the atomic size has increased. If you go to even like a apotin alpha or rituximab or coagulation factor which is used for blood clotting protein, one can see that complexity will increase manyfold as we go from a small molecule based to big molecule like a biologics drug. But nowadays biologics drugs are becoming popular because they are used for treating many of the critical or chronic diseases like autoimmune disorder, cancer and many those where these are used. So, complexity increases, that is what we are saying. As we go from a small molecule based drug to big molecule based drug, complexity increases manifold. Here aspirin which is used for headache 21 dalton, right, and if you go to IgG immunoglobulin 25,000 atoms. So, the complexity increases at every step from production to the formulation to characterizing and matching with a reference listed drug, that becomes paramount importance. However, they are on rapid rise. If you look at the global biological market size from 2016 to 2024, about like it has increased many folds every year. 10% it is increasing, and now it is $394 billion, right? So biologics drugs, like it can be say therapeutics, protein, monoclonal antibody or vaccine, everything is rising. In last 10 years, you can see here from 2009 to 2017, it's exponentially rising, and it's rising everywhere, all over the world, starting from North America to Europe to Asia Pacific and rest of the world. Everywhere biologics now getting major chunk. Therefore, many of the pharmaceutical industry are shifting towards manufacturing biologic drug. They are advancing the field in preparing or the manufacturing the biosimilar which are of robust quality and very high quality. So when we prepare a biosimilar, the biosimilar development program objective based on totality of evidence, means it has to match exactly to the innovator product. If you look at the innovator product, it has an important parameter of quality characterization, non-clinical study, PK/PD clinical study. However, in biosimilar, the quality characterization, analytical characterization is of very much importance, right? So here, if you do the analytical characterization quite robustly, probably you don't need to do like a PK/PD or clinical study on too many samples. Only critical mass has to be done. So in biosimilar, actually this becomes very important to do the characterization. Now typically, the characterization of biologics drug requires many parameters to be controlled, because they are coming from the biotechnology derived, right? They are coming from the animal source or plant source. They are derived using biotechnological tool, right? So any small change in structure can lead to substantial effect, like a binding capacity with the receptor, or they can cause immunogenic effect. So there has to be, like they must be characterized very effectively. So characterization of biologics is for effectiveness, checking the effectiveness. Immunogenicity, as I mentioned, any small aggregate present in the biologics drug can cause huge immunogenic response, so that must be characterized. And also pharmacokinetic profile. And there are various environmental factors that can change the biological product, even a little bit pH or ionic strength, or agitation. That agitation can come during manufacturing, during packaging, during transportation, or freeze-thaw cycle, or even change in the temperature, right? You are manufacturing at one temperature, transporting at another temperature, storing at third temperature, then say electricity gone. And all these can lead to some kind of change in the biologic drug. It can induce the stress in the biologic drug and can change the structure and therefore effectiveness. It can change the aggregation profile, therefore immunogenicity. Therefore, it is extremely important to characterize very well this biologic drug. So typically for characterization of this biologic drug, small peptide or protein, first you have to do the primary sequence analysis. Then one has to look at the physiochemical property, if there is a modification like PTMs that are preserved accurately or not, so that must be done. Then comes higher order structural analysis, which includes secondary, tertiary, quaternary and structure. So together these are called higher order structure. This must be characterized very well. Therefore, like efficacy depends upon how folded or how correctly folded this peptide protein drug are. So that should be ensured that there is no meaningful difference between the innovator drug and your biosimilar, or if you are working with an innovator molecule, that should be ensured that they are correctly folded and thus induce the effective binding with the receptor. Another important characterization is oligomeric state or aggregation state. The impurity can come from anywhere, manufacture related or degradation related impurity. Therefore, biological evaluation of these biologic drugs to illustrate the mechanism of action is very important, and immunogenic response if there is impurity that must be checked, right. So, these are some of the characterization that must be done. Now, for higher order structural, that is the topic of the talk today. Higher order structural characterization can be done by various chromatographic technique or low resolution structural technique. Chromatographic technique for getting the correct molecular weight, you can do the SEC, size exclusion chromatography, helix ion chromatography, the ionic ion exchange chromatography. All of these chromatographic technique can ensure that your two products are same in the elution profile. Then one can use a low resolution structural technique which tells you about secondary and tertiary structure in low resolution, like a circular dichroism, FTIR or Raman. For HOS, one can use HDX, fluorescence. Light scattering gives you idea about the scattering or the globular size of a protein, and then DLS, dynamic light scattering or analytical ultracentrifugation, gives the profile of higher molecular weight species or degraded product if there are in biologic drug. So, they in concerted manner, all of these helps in characterizing the higher order structure analysis as well as aggregation of these products. Now, coming back to NMR, why NMR is exciting? It gives the atomistic picture. So, as we mentioned in the previous minutes, biologics are a complex molecule. Therefore, 2D NMR allows us to separate the signal in a specific region and gives us opportunity to look at the molecule structure in quite detail. The great thing about NMR, it is a highly reproducible. If you are not changing anything, any parameter like temperature, pH or molecule, all the time you record the spectrum without any deviation, you can get a reproducible, exactly reproducible spectrum. So, that is a great thing. Even for batch to batch analysis, this kind of measurement ensures you that even you do not introduce any variation in batch to batch preparation. The only challenge with NMR, it is a low sensitive technique, therefore, it requires quite a bit of samples to analyze. Now, advantage of NMR over other biophysical technique. So, here we are not introducing anything, right. It's without any perturbation. We are not manipulating sample because of high concentration. So, we are neither diluting nor adding any probe, like fluorescence requires a probe or many other techniques requires a probe. Here we do not add any probe. We are not even removing excipients as it is, taking from syringe, recording the NMR spectrum. So, that is a really great advantage. You are not perturbing your biosimilars or biologics when you record an NMR spectroscopy. And these dilution or removal can potentially lead to changes in protein structure or a small peptide based drug. So, other techniques, if you manipulate this, they can change the structure, that is known, right. A structure depends upon the environment. In many of these techniques, you either dilute it, remove the excipients. For doing these experiments in NMR, you do not have to do anything. The other techniques are low resolution, whereas NMR is really high resolution. Only limitation, it is a low sensitivity, right. So, no manipulation in NMR, no change in protein structure, it is a high resolution, that is what we have. So, let us look at what we can do. So, here I will show you the 3 API, right. So, if you just one dimensional proton NMR spectrum, you can see API 1, API 2, API 3, all of them matches quite well. But you see there is a little bit hump here, right, in API 2, that shows although the monomeric profile of these 3 APIs, active pharmaceutical ingredients, we are taking 3 batches or coming from 3 different pharma company, here they are very same. Only thing in API 2 there is a little hump. This could be coming because of the some higher molecular weight species roaming around in the NMR tube, and that may introduce this kind of hump. Otherwise, structurally all these 3 API are very same, right. So, you can see the dispersion in the amide region, which typically tells about the folded structure. The secondary structure seems to be very good, and yeah, and one day so this qualitatively we can say that they are very similar. There is some differences, however, if we want to go for higher structural, then we have to go to 2D. Now, if you look at here, the API formulation 1, formulation 2 and placebo. Placebo basically is added in formulation. So, one can see here these high intensity peaks are coming from placebo. The structurally API and formulation 1, formulation 2 seems to be matching quite well, and whatever this higher molecular weight I was discussing with you in the last slide could be coming from like higher molecular weight in the API, and upon formulation there seems to be gone. So, here you see a flat profile. So, peptide conformation, one can conclude here peptide conformation remains conserved in the formulation state. So, formulation essentially not changing anything in the peptide structure, and the component of placebo or excipients are not interacting with the peptide. That is what one can infer from the one dimensional spectrum. Now, that is qualitatively it is as good as any other chromatographic technique or low resolution, but if you want to go to higher order structural analysis, one has to move from one dimensional spectrum to two dimensional spectrum like a 2D, homonuclear or heteronuclear, where we establish through bond correlation or through space correlation. In toxi, we establish the through bond correlation, total correlation spectroscopy. In NOGI, we establish through space correlation, or one can do a heteronuclear correlation like a amide nitrogen with amide proton. So, here it gives the fingerprint spectroscopy, or you can do carbon proton correlation, heteronuclear. So, these kind of various correlation one can do it. So, if I do say homonuclear correlation on biologic drug like insulin, one can get a beautiful spectrum. So, you can see here the NOGI spectrum giving lots of peaks, which says very clearly that this 51 amino acid small peptide is very well folded, and therefore, we are getting lots of peaks. Now, you can use these peaks to assign and find it out each of the dots that are there, and one can get the structural entity. Now, insulin, as you know that there are 3 disulfide bonds, and that can be also investigated if they are correctly folded or not. So, you can see in NOGI that 2 peaks are coming together, and that says that they are folded, and therefore, if you are getting a peak in NOGI, it says they are within 5 angstrom distance. So, not only the structure, but it also established the critical quality attribute by recording this NOGI experiment. Now, as I mentioned, one can use heteronuclear correlation spectrum. So, at natural abundance where nitrogen 15 is 0.037%, you can record overnight and get a beautiful spectrum of another small peptide drug called semaglutide. This is used for obesity and glycemic index control. Now, you can see each dot here represents one amide correlation, nitrogen 15 with a proton. So, any variation if you see little bit here, one can find it out that variation happening in these amino acids. But these two experiment, toxi and NOGI and N15HSQC, takes a long time. Therefore, quite a few people including we have shifted towards this methyl HSQC or HMQC, what we call, because if you look at the protein structure, there are 6 amino acids that has a methyl, right? Alanine, valine, leucine, isoleucine, threonine and methionine. And if you look at their distribution, they are distributed all over the peptide or protein, like here for insulin, they are distributed all over. So, we can capture the resonances coming from here and then understand about the 3-dimensional structure of a protein or peptide. The good part of this methyl, they give intense signal, they have a favorable relaxation property. So, we use this, exploited this wonderful nature of methyls, recorded the methyl HSQC spectrum less than 2 hours, and you can see beautiful spectrum you are getting here. So, each dot represents one methyl group. So, one can assign these and find it out what are these. So, like here it is coming from isoleucine 2, here it is coming from isoleucine 10, so and so forth. You can assign this using of course toxi and NOGI, and any variation happening in the protein structure one can easily distinguish it. Now, little bit on the formulated state NMR spectrum, right. So, high, like many of these drugs are formulated at a high concentration, right? So there are challenges formulating at high concentration, because at that high concentration there is an instability of a protein, and that instability can come from physical instability. They can aggregate or they can degrade chemical integrity, that can lead to self-association, physical instability, aggregation, phase separation, or degradation can be coming from fragmentation, oxidation, right? So formulation teams spend lot of time to optimize the formulation condition, and they have to continuously assess the formulation and stability. So typically, actually it should be done hand in hand. The analytical characterization should club with the formulation team at every step and ensures that protein structure is not changed. Now, one is manufacturing, right? You manufacture protein at high concentration. Fantastic, you did not find any aggregation, but during the storage or even administration, the aggregation or degradation can happen. Therefore, at every step of biopharmaceutical production, all the way from the designing experiment to formulating to filling the syringe to storing to administration, transportation and administration, actually your structure should be preserved, right? So I will take you an example of insulin and show you, demonstrate you how we can use NMR in characterizing even highly concentrated protein formulation. So if you look at insulin, comes in variants: fast acting insulin, slow acting insulin, intermediate acting insulin, and that is coming because of the structural oscillation that you see in insulin. So insulin generally formulated in a hexameric form, which is stabilized with zinc and protonium sulfate. It's called R6, relaxed state hexamer. Then it can, phenol can or metacresol can exit, and it can go to T3R3 or T6, where zinc can go off, and then you have a dimeric and then monomeric. Now this monomeric actually enters to the capillary, blood capillary, and that is used for controlling the glucose, blood glucose, right? So this is the oscillation happens in regular insulin. However, if somebody requires a slow insulin or intermediate insulin, they form a crystal. Now crystal slowly dissolve into blood, goes to hexamer, T3R3, T6, dimer, and then monomer, and then it enters to the bloodstream. But somebody requires a fast acting insulin like a Lispro and Spart, actually this hexamer is not that stable. So easily it can dissociate to monomer, and therefore supply of insulin is quite rapid. There is a ultra long acting insulin like glargine or detamere. So what they do, they formulate so that it precipitates in the blood and then slowly diffuses. So with the albumin it goes to blood. So depending upon what is the requirement of a person, their insulin are formulated differently, because upon formulation you have a different release profile, like a Spart and Spart and Lispro actually picks up very fast and then they last for 4 to 6 hours, whereas prolonged acting insulin like glargine actually once a day dose is required, and therefore its action is very slow. So now one can see insulin is one medicine which is used for personalized therapy. Depending upon the person requirement, it is formulated high. But insulin, like here NPH, or here you have a microcrystalline or crystalline state. Now how to investigate the crystalline state structure, that is one of the crucial. So typically what people do, insulin act at pH 7.4, but like here it has a crystalline state. But when you have to characterize, typically people acidify it, bring it to the monomeric state, and then do the characterization using HPLC or using any other technique, right? But during the formulation you have a suspension, right? Some can be soluble, some can be insoluble, like a microcrystalline. Now currently what is done, you just shift your equilibrium towards pH 2.3, record proton 1D. Here I show you few of the insulin variants, record proton 1D and proton 2D, and you show that they are similar. So if you look at here 1D or 2D, you can clearly see this region, amide region, each peak matches very well, even in the aliphatic region matches very well. So you are sure that actually it is matching very well, your biosimilar product is very good. You can do 2D, and even little bit variation introduced because of phenol or metacresol you can pick up, right. So any variation you can easily pick up, but you are sure that your monomeric state in this case is perfectly fine. There is no variation. You can even do the spectral correlation analysis like a CCSD that basically takes care of proton, carbon chemical shift and intensity, and you can match two reference product with a biosimilar, and you see if the deviation is low, they are very well matching. You can do the correlation coefficient plotted by this. So using this even statistical analysis, one can show that they are highly similar. The other thing one can do, even go deeper into a statistical analysis, one can do the principal component analysis by taking three or more reference product, three or more the drug product, and look at the variation by removing the excipients peak, taking the well resolved peaks coming from the peptide, and here you can see the separation is quite good. Already we are achieving 90% separation between the RLD and IHDP, and principal component analysis say that these two products are very nice. The another important parameter to be used for this statistical analysis is called Mahalanobis distance. This is a statistical way to show the similarity or dissimilarity between two type of products. So that can be derived from this PCA value, and if the DM is about 3, you are sure that it's very nicely matching, right. Or you can do the this BioHOS echoes developed by Brucker and Mestranova. One can see if the two products are matching very well, you have a nice correlation coefficient. If they are not so matching well, you can have slightly compromised correlation coefficient. So all those are done. We can show the similarity of the product with the reference product in a very coherent manner. In a statistically significant, robust manner, one can establish it. But remember here what we have done. We have shifted the equilibrium from pH 7.4 by acidifying from pH 7.4 to 2.3, which shifted the equilibrium from hexameric state to the monomeric state. Okay. So we will address this little later. Now, one of the problem that we have of biopharmaceutical is is the interference coming from the excipients, right. So here you see on one of the biopharmaceutical that we recorded, here are the excipient peaks, but there are some tricks in NMR. One of them is Sierra filter. If you introduce that with your methyl HSQ6 spectrum, you can really get a beautiful spectrum of where we remove these excipients, and you have a beautiful spectrum, and now this can be used for a statistical analysis, right. So we remove the excipients and we are not perturbing anything. So one can again see how does it orthogonally compare with other techniques. One of the technique used for aggregation analysis is your AUC, analytical ultracentrifugation. So we used insulin at two different concentrations, 1 mg per ml and 2 mg per ml, and we recorded the analytical ultracentrifugation, basically absorption spectrum, and one can see with the increasing concentration the molecular weight or even the sedimentation coefficient does not change, right. So that ensures that your product is remaining in one state rather than going to two state. But this kind of analysis, aggregation analysis, can also be done by a simple trick of NMR called diffusion order spectroscopy. Essentially, DOGE measures actually translational diffusion, right. So it based on, suppose we are starting with a mixture of compound, they basically diffuse differently in the solution, and using something called gradient pulse in NMR, one can find it out how they are diffusing. So one can use for like a small molecule binding to protein or aggregation kinetics or various other effects. So basically what it does, in the tube you apply a magnetic field gradient, and that induces diffusion in the molecule. So you give it some time, and then they will be diffusing with their own speed, and when you reverse the gradient, they will be keep diffusing here, and you measure the intensity of these. So one can measure the intensity with a gradient strength by varying gradient, and one can see this decays, the normalized intensity decays with a gradient, that you can fit it and find it out what is the diffusivity d value. So once you have a d value, one can recall, or we can calculate the hydrodynamic radii of this products using Stokes-Einstein relation, right. So here is K is Boltzmann constant, T is a temperature, this is the viscosity, and pi is of course pi, and R is hydrodynamic radii. So we recorded on insulin analog, and we found D coming to be 1.58 into 10 to the power minus 10 meter square per second, which translates to 1.75 nanometer of hydrodynamic radii. And if we compare with the diameter, one can see it's about 3, so we are getting very high correlation. So we use this technique of NMR to record the diffusion coefficient of various insulin analog, and all of them were coming more or less same in the range between 1.5 1.6 or so. Now the another thing we did, we again combined AUC with basically to look at other small peptides which is liraglutide, and we again recorded the diffusion, NMR diffusion as well as the AUC diffusion, and we found that actually liraglutide shows a heptameric structure. So that's, one can show that NMR can be used an orthogonal technique like any other AUC or so to find it out the structural entity of any biopharmaceuticals. That's so far so good for say a small molecule based drug. Let's move to like a bigger molecule like a monoclonal antibody. So they are really big. If you look at their line width increases as we go from 8 kilodalton to 50 kilodalton, because they are stumbling slow in solution. Therefore, they have a poor signal to noise and there is a severe signal overlap. Now, because of this, they are typically not amenable for NMR. Line width is really broad, there is overlap also, but again we can use some of the NMR techniques for showing the biosimilarity. What we did, we measured this diffusion of formulated biosimilar, like a biosimilar, and compared with the originator, and you can see here the biosimilar hydrodynamic radii coming very nicely, and we also match with a dynamic light scattering, and we published this in Journal of Biopharmaceutical Analysis to show that DOGI can be used even for the maps showing biosimilarity. One can use the one-dimensional proton spectrum and match peak by peak originator biosimilar 1, biosimilar 2, biosimilar 3. One can see here in the aliphatic region, in the amide region, everywhere like more or less the signals are matching. So qualitatively one can see even for MAB that they are showing similarity. And we also, after the experiment, we recorded the gel elution profile to show that there is no aggregation, there is no degradation. So one can see all of them are falling in this region, shows that they are intact during the experiment that we did, right. So, but NMR we don't stop at just 1D and DOGI, we can go to 2D. Now 2D again, what I explained, we can exploit the methyls present in the monoclonal antibody. So this is, remember, 150 kilodalton complex protein, and if we just label methyl here, they are present all over the molecule, right. So in the Fab, in the FC, everywhere. So if we can get a signal from these methyls, we can report about all the important region of a monoclonal antibody, and that's what we did. We elevated the temperature so that it does not aggregate, degrade, and tumbling becomes little faster, and we recorded again this methyl TOGI experiment, and one can see we are getting more than 80-90 peaks coming from this biosimilar trastuzumab, and get a beautiful well dispersed spectrum. So that's giving the signature of this MAB. Now one can really go and do amino acid type of analysis. So one can see here are isoleucine, methionine, alanine, valine, lysine are also shown here, threonine. Lysine should not come, but lysine has CH2, right, that also can be picked up wrongly in this experiment. But you see where this information can be used. Methionine oxidation is one of the critical consideration in biopharmaceutical. So if any shift in these peaks happens, you know that methionine is getting oxidized, right. So one can record NMR spectrum of say oxidized product versus normal product, and you see this peak in shifting in the peak can show the variation in biosimilar product. We can even do the like a correlation coefficient. So we divided this map into different sections, and essentially we match with reference product with a biosimilar product. Show here that they are matching, and one can do the correlation coefficient or CCSD to show that they are similar or dissimilar. So we found it that our biosimilars were quite matching very well. Next, so can we go to more complex system using NMR? So as I was saying, this biopharmaceutical, many of the biopharmaceutical are formulated in microcrystalline formulation. So typically for characterizing them, people either dilute it and shift the equilibrium towards monomer by acidifying it and break that. But we thought we should not perturb anything. Let's do as it is. Now they are not soluble. So what we can do? So NMR has another cousin called solid state NMR, which can be used for microcrystalline suspension. And for soluble one can use for biophasic. If we are working with a biophasic, we can extract the soluble part as it is recorded spectrum. We can centrifuge and get the microcrystalline part. We can record that spectrum as well, and that's what we did. So we like a sedimentation or frosty, it's on edge of solid state and solid state. You can pack your biopharmaceutical in rotor, spin very fast. All the molecule will be sediment on the wall, and then you can record the spectrum beautifully. So here one can see here are my molecules are sedimenting, and you can really record a beautiful spectrum. So here that's what we do. We centrifuge it, pack in the rotor like this, and spin at very high speed like a 30 kilohertz or so, and then you show the similarity between various formulated state like 1 versus 2 versus RLD and all those, and you see even from the microcrystalline substance each peak matching very well. That shows that gives us confidence that directly from the informulated state one can take this biopharmaceutical and show the similarity. We can record a 2D proton detected carbon 13 proton correlation spectrum biosimilar versus RLD. They are also showing very high similarity, right? So one can conclude here, like here I showed you not only 1D, we can do 2D, and one can again go ahead and do the correlation coefficient or Mahalanobis distance or PCA to show how similar and dissimilar it is. So with this, I was able to convince you that NMR spectroscopy is an excellent tool for biopharmaceutical application. I showed you in the initial part that methyl HSQC can be used for showing biosimilarity or higher order structural characterization in a robust manner. Next, I showed you that diffusion order spectroscopy DOSI, which gives the size of a molecule in the native state, can be used to understand the aggregation property or degradation of a biomolecule. And if you combine that with analytical ultracentrifugation, even you can detect the smaller fraction of aggregates. So DOSI and AUC can go hand in hand for characterizing the aggregation or degraded state of various biopharmaceutical, or batch to batch analysis can be used using proton NMR in a quicker and simpler way. Next, I move to solid state NMR, and I showed in fact we can do the higher order structural similarity of drug in formulated state without perturbing anything, directly taking from the pen. You can record the solid state NMR spectrum, and I also showed that we can even tackle the monoclonal antibody, which is high molecular weight, and in a qualitative as well as quantitative manner we can investigate the higher order structural analysis. We can investigate their aggregation state or degraded state, various aspects. So I can, in summary, I can say that NMR spectroscopy is fabulous tool for biopharmaceutical analysis, and I think it should be adopted very well by biopharma industry to have a robust characterization of biopharmaceutical manufactured product, stored product, right, even during transportation up to everywhere. it should be used for characterizing it very well, because its life matters, right. So, this drug has to be characterized very nicely. It is something to do with the human life. So, robust characterization is always helpful in giving confidence of whole community that drug that we have manufactured or drug that we are administrating is really excellent quality. So, with this I would like to thank various collaborators who gave me sample for doing these analysis. So, we have worked on various biopharmaceutical that you can see, teriparatide, liraglutide, glatimer acetate like cetrorelix, and liraglutide, semaglutide, insulin and all those. And yeah, and we combined various other bio orthogonal techniques like AUC, SECMALS, ITC, DSC, LCMS, MALDI, SPR and all those, and I collaborate with various pharma companies like which is there. And at the end I would like to thank my group members. We work together as a team, try to solve some of the problem in structural biology as well as biosimilarity, and IIT Bombay for funding this instrument, and also other funding agency for funding my lab. So, this work was mostly done by these three champions, Soumya Ranjan Pujahari, who is a PhD student, Rudra, who is working as a postdoc in my lab, he developed methods for solid state, and Pramod, he works also on biopharmaceuticals, he takes care of magnet, and I am really thankful to this big magnet who helped me in generating data and knowledge. So, I am thankful to all of you. Thank you very much. With this, I would like to close. Have a good day.